Okay, good afternoon, everyone. I'm Jane Wall, Director of Business Development and Membership at BIA. Welcome to our BIA webinar on innovative partnerships in dementia research, which has been pulled together with the help of our charity partners for 2017, Alzheimer's Research UK. Just a couple of notes of housekeeping. Everyone is currently muted in order to avoid background noise. We will have a Q&A session at the end, so please do leave your questions in the question box, which can be found on the control panel towards the right throughout the webinar. Please remember to state who the question is for, and we'll take those at the end. Just for information, this session is going to be being recorded and will be uploaded to YouTube tomorrow. So if you found it useful, please do send it on to colleagues and others. So we're privileged to have with us today Dr. David Reynolds, Executive Director and Chief Scientific Officer of Alzheimer's Research UK, Dr. Janet Brownlees, Team Leader at MRC Technology, Dr. Rick Livesey, Chief Executive Officer, Gen2 Neuroscience, and Dr. Lee Dawson, Vice President and Head of CNS at Astex Pharmaceuticals. So without further ado, I'm going to hand over to Dr. David Reynolds, Executive Director and Chief Scientific Officer of Alzheimer's Research UK, to provide an overview of the dementia research landscape and funding models that have been developed to increase investment in drug discovery. David, over to you.
Thank you. First of all, apologies for my slightly croaky voice. I'm recovering from a cold, but hopefully I will be clear enough to everybody listening. So I thought I'd start with a little bit of background on the impact of dementia and where we are in this field before we then look at actual drug discovery activities, as I'm aware that not everybody will be an expert on dementia. So if I go to my first slide, in terms of the impact of dementia in the UK, there's about 850,000 people living with dementia, and that number is predicted to grow to 1 million by 2025 and increase further to 2 million by 2020. And this is a picture that's seen across the globe with an aging population. The number of people living with dementia is increasing. The costs to the economy of dementia are actually very high, about £26 billion a year. And given our current predicted lifespans, 1 in 3 people born this year is expected to develop dementia at some point in their life if we don't do anything about either really understanding ways of preventing and reducing risk or indeed being able to treat dementia. Now, the impact of dementia is actually much higher than it is for several of the other major diseases. And in fact, the health and social care cost of dementia is higher than cancer, stroke, and heart disease combined. And yet, if we look at the number of researchers working in the dementia field, there are less than in any of those 3 areas individually. Now, things are improving, and in 2008, there used to be 1 dementia researcher for every 6 cancer researchers, whereas if you look at data from sorry, there's a bit of a delay on getting the slides to advance. If you look at the data from 2015 based on publications, there's actually 1 dementia researcher for every 4 cancer researchers. So we're definitely moving in the right direction of increasing investment in this area. Now, what does that yielded us so far? So I've done here a timeline of where we are with understanding the disease in orange at the top and where we are with treatments in the purple at the bottom. So although we've known about Alzheimer's disease for over 100 years, it's only been since about the late 60s, 70s that we've really understood that it's a disease that's caused by neurodegeneration. And then it's only from the 90s that we've had genetics to help us start to unpick key disease processes. And then in more recent years, over the last 5 to 10 years, we've understood a lot more about the genetic risk factors for the disease and the key cellular processes that likely underpin the development of not just Alzheimer's disease, but many of the neurodegenerative diseases. Now, on the therapy side of things, we're not doing very well when you compare to many other disease areas. In the late 90s, there were a number of acetylcholinesterase inhibitors launched. So these pep up your cholinergic system and provide symptomatic benefit for a short period of time, usually up to about 2 years out of a 10-year disease course. But they don't have any impact on the disease progression itself. In 2002, memantine was launched, which is another symptomatic treatment affecting the NMDA glutamatergic system. And that's been a bit of a kind of orphan drug in that there haven't been any other similar molecules in that class launched. Now, a lot of effort has gone into developing disease-modifying therapies for Alzheimer's disease, and an awful lot of things have failed over the last 15 or 20 years. And most recently, an amyloid antibody called solanezumab failed its phase 3 trials in mild Alzheimer's patients at the end of last year. And even, I think it was last week, that Merck gave up on their mild to moderate patient population for their BACE inhibitor. They are still progressing forward with other studies in different patient populations, the prodromal, so the patients that haven't yet fully developed Alzheimer's disease, in the hope that we can prevent this disease progressing. But we don't know when we're going to get disease-modifying therapies through to Alzheimer's patients. And for other rarer forms of dementia, there's very little on the horizon. So, why is this? Well, for this audience, For this audience, I don't need to go through the drug discovery and development cascade. Rather, what I want to point out is why, in this area, it's much longer and harder to develop drugs. And it really comes down to two things. One, we have a very limited understanding of what causes the diseases, and therefore our choice of drug targets is perhaps not that well informed. And our clinical trials are longer and much more expensive than many other areas because the disease is relatively slowly progressing and somewhat uneven in its progression. So you need to have trials that are quite long to be able to see a drug effect. And our clinical endpoints, various forms of rating scales, are pretty blunt instruments with a lot of variability, which means you need a large sample size to be able to detect robustly a change in a rate of decline. This means that it's long and expensive to develop drugs in this area, and so far nothing has made it to the finish line over the last 15 years. So what are we doing about it? Well, Alzheimer's Research UK, I think, has this quote here from David Cameron, kind of positions us. We're in the kind of the right place, that intersection between good quality academic research and then linking that through to our cutting edge biopharmaceutical industry. And a global initiative on dementia was launched by David Cameron when he was the Prime Minister back in 2013, and he recently became the president of our charity to help carry forward that commitment. Now he's no longer in government. So I put here the gaps in the dementia landscape, which I've kind of really covered to a large extent. But one of the, in the primary causes section, the high failure rate and high cost of R&D in this area has meant that many companies have withdrawn investment from this area, and many have pulled out altogether. And they're rather risk averse to go back in and put further investment in. And that, I think, is quite understandable given the economics and the likely return on investment. So as a charity, what we're trying to do is help drive that basic understanding of what's causing these diseases, and then help increase the number of validated targets, because we know that's the point at which biotech and pharma are keen to pick up and run with a new target and develop the drugs of the future. So in terms of the models that we've set up at Alzheimer's Research UK, we've really thought about 4 different ways of doing this, and we have a different role in each one. So starting on the left, we set up a dementia consortium, which Lee will talk about later, where it's a consortium led by us with the MRCT, working with a number of different companies. We've also set up the Drug Discovery Alliance, which I'll talk about a little bit more on the next slide. The Dementia Discovery Fund was initiated from the Prime Minister's Global Challenge on Dementia, where the government put in £15 million into the fund, and then through a number of farmer partners, six farmer partners, there's a fund of about £100 million that's designed to fund dementia projects in a VC manner, because not enough investment goes into this area, and Rick has been one of the beneficiaries of that fund, and we'll talk about his experiences there. And then at the bottom, we have a charity and MRCT-led consortium where we're trying to reactivate projects that have become stalled in large pharma and get them going again to move them down the drug development pathway. And of course, all of this is underpinned by basic research, some of which we fund through investigator-led grants, and the Dementia Research Institute, this large £250 million investment that was announced in December, is key in kind of driving and accelerating our understanding of these diseases. So just briefly, our Drug Discovery Alliance, we've set up 3 institutes, one in Oxford, one in Cambridge, and one in University College London. And we've funded them to the tune of £10 million per institute over a 5-year period. And the aim here is to bring together the academic world-class disease biology in those universities and couple that with industry know-how to build target validation packages. So really to help de-risk that academic science into targets that have hopefully got legs for industry to work on. And the key thing here is it's not academic drug discovery. It's professional drug discovery in an academic environment. And by that, what I mean is each of the 3 institutes is led by highly experienced industry CSOs with industry experience leads in biology and chemistry. So they, as a leadership team, know what a good validation package looks like. What does a biotech company or a pharma company want to see about a target, the reagents and assays that are available, and the various other pieces of information that de-risk that target to drive it forwards. And that's what those teams are doing, working closely with the world-class academics we have in this country, particularly in those 3 institutions. And then this is just a snapshot of the portfolio. And really, what I wanted to indicate here is that our 3 institutes work as a collective alliance so that the portfolio is integrated across the 3 different institutes. Each has its own area of scientific interest, but they complement each other. And we've had a lot of interest from industry. In 8 of our projects, we've already got some element of industrial partnering with them, and that's across 5 different companies. It's early stages for the Drug Discovery Alliance. We really started our wet work about a year ago, and the projects are starting to make good progress. And so that's all I'm going to say. And really what I want to do now, if the slides had found, is hand over, first of all, to Janet, who will talk about the Dementia Consortium, and then to Rick, who will talk about his experience with Gen2 as part of the Dementia Discovery Fund, and then finally to Lee, who is involved in all 3 of the approaches we have. So over to you, Janet.
Okay, thank you, Derek. So as David says, I'm going to talk a little bit more about the Dementia Consortium. How to get these slides?
There's a delay on the advance.
Down here. Sorry, slides are stuck. So I'm going to just cover how the Dementia Consortium started, why it started, where we are now with it, and the plans for the future. Oh, there we go, thank you. But first I'm going to give a little bit of background on MRC technology. So we are a not-for-profit medical research charity that sits between the academic and the pharma sectors. We form partnerships and collaborations to drive early-stage scientific research to a point where pharma or biotech will be interested in a licensing deal and progress the project or lead molecule into the clinic. We did spin out from the MRC in 2000 as a technology transfer organisation, but we're now quite separate. We have our own dedicated purpose-built drug discovery laboratories in the Centre for Therapeutics Discovery, and we work on both small molecule and antibody therapeutics for all disease areas of unmet medical need, and we have a track record of success in getting drugs to the market. So within the Centre for Therapeutics Discovery, we have capabilities to support early stage drug discovery activities, including compound libraries, assay development, cellular pharmacology, medicinal chemistry, structural biology, antibody engineering, and ADME. And as mentioned earlier, we have success in delivering new drugs to patients with 4 drugs on the market for cancer, MS, Crohn's disease, and rheumatoid arthritis. A further 10 are in preclinical or clinical development. So the Dementia Consortium is a new partnership to translate fundamental discovery in neurodegeneration. It started in 2014 in response to the G8 London Summit and to global concerns around dementia and, of course, the predicted very high cost for societies. So the purpose was to bring together charity, academic and industry sectors to investigate higher risk novel targets and to accelerate novel therapies. So it is a collaborative risk-sharing partnership to de-risk novel research, be it target validation or drug discovery projects. There is a need for such partnership as it can be tough to get grant funding in academia and for drug discovery, and also pharma have largely withdrawn from early stage research in neurodegeneration, as David's already mentioned. So there is a gap which definitely needs to be filled. So how does the consortium work? There are 4 groups in the consortium: AR UK and pharma partners listed there, AbbVie, Astex, Eisai, Lilly, and Merck Sharp and Dohme, provide shared funding for the projects and also come with disease and drug discovery expertise. The academic brings knowledge in novel biology, and MRC provide the drug discovery, target validation capabilities, and management of the application process and active projects. So the application process, projects are recruited globally either through networks or active scouting at academic institutions or conferences, or by direct approaches from academics through the website. There is a 2-tier application process. First, a 2-page outline proposal, and if successful, this will progress to a full application. The approval decision is joint between AR UK, pharma partners, and MRCT. Once approved, the pharma partners at this stage declare if they want to invest, and those that don't drop out at this stage. And then there is a more detailed, milestone-driven project plan and costings drawn up based on further discussions. A collaboration agreement is signed and the feasibility phase is started. So the feasibility stage is usually milestone 1 of the project plan, and there are clear no-go decision points incorporated into the plan. So all results are shared with all parties, and reagents are shared between the academic and MRCT. So just a brief word on the project IP. The funding industrial partners for a particular project can use the results for in-house research under non-exclusive license. If they want to take it further into full drug discovery project, then they can negotiate an exclusive commercial license. Where there are 2 or more funding partners, the options can be co-exclusive. So this is a timeline for partners joining and projects starting in the consortium. So at the left-hand side at the top, the consortium was launched in March 2014 with 2 companies, Lilly and Eisai. 3 other companies, Astex, AbbVie, and MSD, joined at later dates, as can be seen along the top of the timeline. So the funding was for $4.5 million over 3 years, but this has just been extended by another year. So our first project started as the CSF1R project from the University of Southampton, and that started in June 2015. And this had paved the way with the establishment of the processes and the processes to get agreements in place, and the other 4 projects started quite quickly after that. The projects are from various countries, 2 from the UK, 1 from Italy, 1 from Luxembourg, and 1 from the US. There are further 2 projects currently going through the process. So the advantages and challenges of such a consortium, working with a disease-based charity and with PIs brings a deep understanding of the disease area and novel biology. The perceived challenge may be bringing the academic and industry interests together. But actually, as all parties are involved in open collaborative discussions around project selection and planning, bringing the differing viewpoints together has not actually been that difficult, although it does take a few iterations to get the plans agreed by everyone. And of course, these plans may also change based on the milestone-driven results. One challenge for the academic may be that the milestone-driven funding could cause employed staff uncertainty, as it's not completely predictable how far or for how long projects will run. And there are basic logistical challenges when trying to get so many busy parties together. The partners need to be flexible and committed, which indeed that has been the case, and everybody seems very committed to make this consortium work. So what's next? We have had 80 applications since 2014. 10 progressed to full applications, and that does appear to be quite a drop, but many of those initial applications were out of scope for the consortium. Many were repositioning, which actually fit better under other funding or company-funded processes. So there are 5 active projects launched, and one's in feasibility and one's currently under review. So what does success look like for these projects? Well, ideally, it's a validated or a devalidated target, or the initiation of a drug discovery project. But they're all still in the early stages. All of them are progressing well, but we haven't reached a stage for decision points or decision points to terminate on any of these projects as yet. The agreement runs to March 2018, and we are continuing to review new projects. So the need for such a consortium and risk sharing in the neurodegeneration space is recognised, and the continuation of the consortium is in the planning stages. And I would say things to consider going forward are the sourcing of projects, for example, by more direct approaches to centres of excellence, not just in the UK, but across Europe and the US. And perhaps working at even earlier stages with academics, for example, providing tool compounds, libraries, or in silico structural support to enable a more robust data set and higher confidence in the target or pathway. It may be possible to recruit further former parties, but perhaps not feasible to have too many, as there's probably a critical mass that works well, and too many may make it unwieldy and unworkable. So in conclusion, we would hope that the consortium will be successful in initiating drug discovery projects which will help the field move forward towards the goal of treatment by 2025. And just to finish with an acknowledgement slide, and these are the main people involved from the different organisations, and there is a contact email address. So thank you, and I'm going to pass now to Rick.
That's to Rick.
Yeah.
Afternoon, everybody. My name is Rick Lipsey. I'm the CEO and scientific founder of a company that was funded by the Neurotech Discovery Fund called Gen2 Neuroscience, based up here in Cambridge on the Bibram Science Park. So my brief was to give sort of a background of what it's like to be sort of at the user end, as it were, of these things. So I thought I'd give a little bit of background as to sort of the rationale of the company, a little bit of science, and then how we grew out of it. Because in sort of my other day job, I'm an academic in a Wellcome Trust Centre here in the university in Cambridge. So Gen2 is focused on a particular aspect of dementia, which is this emerging finding over the last say 5 to 10 years, that in addition to dementia affecting sort of individual neurons, it also spreads through the brain. And there's a number of sort of models for how this works, but one of the key ones seems to be that disease neurons release toxic forms of a protein which is normally found in healthy neurons. It's an intracellular protein which is part of the architecture of the nerve cell, which in disease gets released by the neurons and then actually spreads the disease from cell to cell. And a number of pharma companies are working in this space, saying, well, it's a toxic gain of function in the disease, so let's target all of the form of this protein, it's called tau, that appear outside neurons. We had found that actually healthy neurons release large amounts of a non-toxic form of this protein. And there's a basic sort of physiological mechanism by which neurons both release this protein and take up this protein. And so Gen2 was founded essentially to target the toxic forms of tau, specifically to slow disease spreading. Because as you can imagine, if there's an awful lot of the sort of normal healthy tau, then you've got a huge targeting problem in terms of trying to hit the toxic form. So to tell you a bit of science, tau exists in 6 isoforms in neurons normally, and there's lots of post-translational modifications. There's historically been a lot of interest in phosphorylation, but that hasn't turned out to be particularly successful as a therapeutic strategy, trying to change the phosphorus status. More recently, there's a lot of interest in acetylation. But what we're showing here on this, these are just pulling down the tau that comes out of neurons and asking what types there are. And what you find basically is, beside the asterisk, is this ladder of different sizes of tau, most of which have the right-hand end, the C-terminus, chopped off. So it turns out, this is highly reproducible, so it turns out that neurons release an awful lot of this. So the other arm of this is that neurons also efficiently take up tau, and this was quite a surprise. And what's shown on the left here, this is still from a movie that hopefully will start in a second. And these are human neurons that we make from stem cells sitting in a dish. And what we've done is we've taken the tau protein, which normally lives inside the neurons, and we've labeled it with a dye, so it only fluoresces when it hits low pH environment. And a low pH environment is when things get endocytosed into cells, so the pH drops. And you can see here on the movie, hopefully, it'll play. No, we give up. I'll try again. I'm being waved at here. Oh no, there we go. Yeah, it might play. You never know. They'll go red, basically. If you anyway, take my word for it. The neurons go red. Honest gov. And the other point of this slide is actually then, so one of the strategies people are following is, well, will antibodies prevent this uptake? And you can see here, the graph on the right simply shows that as you start using different monoclonal and polyclonal antibodies, you can reduce the amount of tau that goes up into neurons. There are some complications to this, which become interestingly both scientifically and therapeutically, but broadly speaking, one can slow down as long as one targets the antibody to the right place. The problem is, this will be affecting all of this normal homeostatic physiological process in people's brains, hence the need to target toxic tau. So what Gen2 is about is, in collaboration with our sister company, a company called Townsend Therapeutics, where we're identifying disease-specific extracellular forms of tau, and this uses human stem cell models of different genetic forms of dementia. We then have lots of assays for the toxicity of these different forms of tau, including electrophysiology, neural transfer, and so on. And the last arm then is to develop therapeutics to block this specific toxic form of tau. So that's the science side. So just on the other hand, how do we kind of get here? So rather than this being my normal acknowledgement slide, this is to remind me that in my normal day job, I work in a thing called the Gurdon Institute. And the Gurdon Institute is a Wellcome Trust Centre in the University of Cambridge. Within that, we actually also run something, as you can see here, called the Alzheimer's Research UK Stem Cell Research Centre, which essentially had built on our original work developing in vitro human neuronal models of dementia. And what that was we essentially discovered ways, a number of years ago, now a movie plays, that's sod. So where we can take stem cells, or we can make stem cells from any individual and turn them into forebrain neurons, human neurons in a dish. And what we're showing in the movie here is we can watch them firing, and we can actually control that firing. That little flash of red followed by the flashing was us actually controlling the neurons using optogenetics. So that's nice. So we can make the part of the brain in the lab that's affected by dementia. We can also make the immune system of the brain, which is very important in terms of dementia. These are these things called microglial cells, which are these guys here. And we can put those together to essentially reconstitute the different elements of the central nervous system, particularly paying attention to the parts which are involved in dementia progression. So, for example, there are human mutations which affect genes encoding proteins which are specifically found in the microglial cells, and there are other ones which are affected in neurons. And what that allows us to do is go from some genetics to disease biology. So far, so academic, and that's all good. So from there, we started this company I mentioned called Talisman, which over 3 years ago, which grew out of collaborations we had with pharma and biotech. And essentially what that is, it's a target validation company which does both internal and external projects. So the pre-drug discovery stuff, working with tool compounds for target validation. And that's privately held. And then I guess more than a year ago, we came up with the idea of Gen 2. And Gen 2, as I mentioned, is designed to develop therapeutics to tau. And that then we approached the Dementia Discovery Fund. So Gen 2 was the DDF's first UK investment. And that's been essential for allowing us to do the first 18 months where we actually do prove the concept behind the company. And what we found with the DDF is that they're much more than a simple venture funder, because they have quite a strong scientific basis. The CSO, or as it were, Tetsu Maruyama is ex-Takeda with obviously an awful lot of pharma experience. So they're quite a supportive organization, both in terms of evaluating the science at the point of spinning out the company, but also in terms of the to and fro of discussing progress and milestones, and as we're finding now, making introductions to talking to people about Series A, for example. And so we've really found them kind of a pleasure to work with. And I'll stop there, and later if people want ask questions about the whole process, I'll be more than happy to answer.
Okay, thank you very much, Rick. We are going to move on now to Dr. Lee Dawson, Vice President, Head of CNS at Aztec Pharmaceuticals. Lee, can you hear us?
I can, yes. Can you hear me?
Yes, I believe we can. So straight over to you.
Okay, so I should have control of the slides, is that correct?
Indeed.
Okay, brilliant, thank you. So we were asked to take part today as one of the pharma partners that spreads across the AI UK network. And we'll go through a little bit of background on Aztecs, where we've come from and what we do. Neuroscience actually is a new area for Aztecs, and actually, unlike a lot of pharma, we've actually moved into neuroscience much more. But we are fueling that largely through a highly collaborative operating model. Sorry, the slide's not moving. Oh, there we go. So just a little bit of potted history on Aztecs for those that are not familiar with the company. It was a biotech that was established probably 15, 16 years ago now by Harun Jyoti and a number of academics, so Tom Blundell, Chris Abel from Cambridge. And it was basically set up around the idea of fragment-based drug discovery and rational design of molecules based on the structure of interaction with proteins. About 3 years ago, we were purchased by Otsuka Pharmaceuticals, which is one of the large Japanese companies. And they've actually invested quite heavily in Aztecs and has actually been behind a lot of our investment in neuroscience. We've really been a pioneer of fragment-based approaches to drug design, but because Aztecs has come from quite a biotech background, the way we work is highly collaborative. I guess it's the technique that's become known as open innovation, but it's very much a finding synergy and partnership and working with pharma partners, charities, academics, and you can see there a list of some of the interactions that Aztecs have had in the past. And we're probably best known for our oncology portfolio, and we have a number of molecules going through various oncology indications in clinical development currently. So Aztecs' platform has become known as Pyramid. So what this is basically highly technical assessment of protein structure, and we then target those protein structures with very small fragment molecules. So these are typically less than 250 molecular weight, which is obviously much smaller than conventional drugs. And these actually interact with the protein. And we use a whole battery of complex biophysical screening technologies which is way beyond my background, to actually detect this binding, optimize those protein interactions, and then rationally design molecules to optimize those interactions with the protein of choice. So what this does and what it offers, particularly for neuroscience, is a way of actually controlling the size and the biophysical properties of the molecule you design. So sometimes, not always, you are actually able to develop molecules which may have optimized CNS penetration. So our neuroscience, and particularly our neurodegeneration portfolio and strategy going forward has largely focused around this technology. So what that actually allows us to do is actually focus on specific targets, but we need to be able to crystallize those protein targets. So we look for pathways and targets that have strong biological rationale to the disease. Ideally, we would then be able to develop biomarkers. We've come from an oncology background where companion diagnostics is part and parcel. That is a long way off for neuroscience, granted, but it certainly is making some progress towards that. So we target those specific proteins that interact within these pathways. Neuroscience, and particularly neurodegeneration and Alzheimer's disease, there's a lot of genetics emerging now to point around key pathways that may be implicated in disease biology. And these pathways have various intervention points and proteins which may be amenable to our technologies. And that's basically what we're looking for. We then employ our pyramid strategy to actually try and find new molecules that will modify the function of these proteins. As I mentioned, we work largely through open innovation alliances. In fact, we pretty much work solely through open innovation alliances. We do have a small team of neuroscientists, but what we do look for is networks, partners, charities, experts in these pathways that we can actually interact with and bring what skills we have to bear on a problem. So we are looking for disease-modifying agents in particular. Sorry, there's a delay in the slides. There we go. So as David mentioned, there's been a number of initiatives set up by Alzheimer's Research UK. These have been a fantastic opportunity to bring our complementarity and our skill sets to bear and to partner. You've heard about the Dementia Consortium and the Dementia Discovery Fund and the the Drug Discovery Institutes and the Drug Discovery Alliances. And Astex actually are involved in all three of these. I mean, what is the advantage to Astex to be involved? It very much enhances our global open innovation networks. It allows us to talk to the academics who work within these interactions and these networks to actually be partners and actually bring their ideas forwards. The Dementia Consortium, as you've heard, brings forward validated targets, such as validation is in a preclinical arena, and those targets may be amenable to our technologies. You've heard about the Dementia Discovery Fund from Rick. This allows us to invest very early with funds. Obviously, this is a venture fund, but also it may expand our networks there also. And it allows Astex's skills and knowledge to be applied to the advancement of the basic science. So we do invest in much of the basic science in partnership to ultimately try and find new targets, which we can then validate potentially as therapeutics for the future. I mean, you've heard from David that the questions that we're asking in terms of neuroscience and Alzheimer's disease are very complex. The biology is very complex, and finding new drug targets is proving challenging. So I'm a big believer in that the strength that we have and our best chances of actually prosecuting some of these targets towards a therapeutic endpoint, so we actually can treat patients, is in cross-sector, multidisciplinary networks where we can actually bring our synergies to bear on these questions and hopefully develop molecules in that manner. So I will leave the Astex there, and if there are any questions, obviously I'm happy to contribute.
Okay, thank you, everybody. Thank you, all of the contributors. Really fantastic example of how charities, academia, and industry working together can build new models of innovation and funding. We're going to take some questions now. We have one, but if anyone else wants to submit some questions in the meantime, please do so. Alexandra is asking if slides will be shared after the webinar. Yes, absolutely, and also, as I said before, the presentation will be on YouTube for you to replay and share. Are there any more questions for any of the speakers? Okay, yeah, we have one more. We've got Denise. Some of you mentioned you are looking for defined targets. What do you mean by defined targets?
So perhaps I'll take that one. It's David Reynolds here. So one of the challenges is that there's a lot of good, really good quality academic research comes out implicating a particular, say, a kinase or a protein or a pathway. But then there often isn't the level of follow-up that is needed to encourage companies to come in and start working on those pathways. So one Nature paper does not mean that it's a great target. First of all, you need to see if it can be replicated. There's important pieces to understand about, is it a druggable part of the pathway? And in fact, in many of the targets that we're finding in, say, our Drug Discovery Alliance that we really want to work on is about disrupting protein-protein interactions, which is doable, but classically a tough drug target, at least for a small molecule. And if it's intracellular, using the likes of antibodies, for example, can be very difficult. And then there are other questions about, is it safe to target that particular part of a pathway? Are there safety implications for in all manner of ways. So those are the kinds of things that really you want to have available, that information available to you when you go looking for drug targets to start working on. If you're in a biotech company where you're perhaps only going to work on a very small number of targets, or in a large pharma where you want to have confidence that your early exploratory projects will lead through to candidates and ultimately through to clinical trials and drugs on the market. So we see the role in Alzheimer's Research UK of kind of trying to bridge that kind of chasm of uncertainty between a great Nature paper and something that is a target that's got legs to become a drug of the future. And that's where our Drug Discovery Alliance or the Dementia Consortium are willing to put that very early stage investment in to get those good tools to try and show that the kind of target does what it says on the tin. Because often academic research, it uses viral vectors or genetics to manipulate a pathway, which is great and it's very flexible, but you can't do that very easily in a human being. You've got to do it with a pharmaceutical agent from description, and those two things don't necessarily give you the same result. So it's trying to de-risk some of the science and the elements we know will become important later in development to get that target that we think industry will be interested in. I don't know whether you want to add to that, Lee.
Yeah, I mean, that's exactly it. We look for strength in the biology that actually intervening at that particular target level will modulate the biology in a manner we feel may be potentially therapeutically beneficial. We're not looking for proof that it will cure dementia, because obviously that's not feasible, but it's just adding the actual story to make the biological story much more credible, I guess, and something that we'd want to invest our capabilities in actually prosecuting.
Okay, thank you very much. That's a very comprehensive answer there. Has anybody got any more questions? No, it doesn't look like we've got any more coming through at the moment. please do send any afterwards if you think of them, and we will try our best to get them answered for you. So thank you once again to David, Janet, Rick and Lee for taking part in this today. I think it's been absolutely fascinating. BIA work on behalf of organisations in the innovative life sciences, and we can't do this without our members. So if you're interested in getting more involved, please do get in contact. Just a couple of upcoming events from the BIA to watch out for. Breakfast with the BIA in Manchester, if you are up that way, on 16th of March. Also, our UK CEO Investor Forum, information just gone up on our website about that. Do put it in your diary. And of course, the BIA MHRA conference is in for the 8th of June. So do visit the website, have a look at the events coming up, and try to get along to some if you can. Thank you very much for joining us. Any feedback, always welcome for the webinars, for topics, for ongoing conversations. And thank you very much, everybody.